to talk about something that I'm not an expert on. So I'm going to try to present my thoughts on reproducibility from the point of view of somebody who's in the lab doing experiments and trying to do the best possible work we can, reading all this literature on reproducibility. I'm probably going to say a lot of nonsense, but it's probably the interesting thing from our perspective is what can we learn from this literature on reproducibility. So I'm also going to try to make this a little bit interactive because it's the last talk of the day. So if you could please take your smartphones and scan that QR code and leave the browser open because I'm going to be asking a few questions during the presentation. Not that I have any good answers. So leave the browser open. So, I mean, honestly, I don't have an expert on this. It might sound like I have an imposter syndrome. It's not a syndrome. And so let me present myself, so where I come from and how we got to this issue of reproducibility. So I am a geneticist. I work with C. elegans. I'm interested in particularly asymmetric cell division. I work in a few other topics like metabolism. And this is the things I work on and I want to do experiments on and try to understand. But I also have a personal interest on how we do science. Since I started the work on science, I wonder what is this job of a scientist and how can we do science to produce robust discoveries. And because of that, when we started our program on cancer biology, graduate program in cancer biology, master's here in Bordeaux, I took that interest into trying to, and I created a module on experimental design. So some of my students are going to recognize a few of the slides I'm going to present today. And so a bit of what I'm going to talk about today is also influenced by my experience in teaching and trying to ask students, talk to students how we do experiments in the lab. So, I'm going to start with just a few definitions. We're talking about reproducibility. There's a lot of discussion about what the definitions, what we mean by reproducibility. This is the one I'm going to use today. It might be a topic for discussion afterwards, but it's borrowed from Lorena Barber's definition. And so it's repeatability. It's when same person or the same team, it's doing the same experiment with the same methods and techniques. I'm not going to talk too much about that today. I assume that everything that is published is at least repeatable. I might be naive, but we're not going to talk about that today. So it's going to be talking more about replicability, which is trying to do the same experiment with the same protocols and same reagents and techniques in a different lab from the original one. As you've seen today in previous presentations, replicability, it's doing a perfect replication of a previous experiment. It's definitely not easy. At best, we can replicate the parameters that are important for an experimental outcome. And then reproducibility is when you do test the same hypothesis, doing a slightly different or more or less different protocol and experimental system that allows you to reach or not the same conclusion. But in practical terms, it's almost always reproducibility because you can perfectly replicate the results. But there's a hierarchy in these definitions. If it's not repeatable, then replicability, it's a moot question. And if it's not replicable, then reproducibility is also a moot question. But there's also the idea that what is reproducible, it's more robust. So that if you can, by slightly different means, test the same hypothesis and that holds, then that's going to be more robust. Where something that is replicable, but it's not reproducible, it's going to be dependent on very stringent experimental conditions might not be generalizable. And mostly what I have in mind when I'm talking today, it's we in the lab testing hypotheses, trying to establish causality effects. So here A causes B, that's the question. So usually, very schematically, we have an experiment where we compared a control condition with the treatment. The treatment should affect A, and if we see a difference in B when we compare control versus treatment, then we consider that there's an effect, and if there is no difference, there's no difference in between control and treatment, there's no effect, we don't reject the null hypothesis. So you might recognize this kind of table. This is the result of the experiment. Okay, this is not the truth. It's what comes out of the experiment, and based on those results, you reject or you don't reject the null hypothesis. Whether it is true or not true, it's what we would like to know, ideally. When we don't reject the null hypothesis, and the null hypothesis is true, then that's a true negative result. We find if we reject the null hypothesis and the null hypothesis is false, then we consider this a true positive, and this is what we would like to have. And the other possibilities, type 1, type 2 errors, we talked about before, that I'm calling false positive and false negative. Here, you don't reject the null hypothesis when it is false, it's a false negative, and you reject it when actually the null hypothesis is true, it will be the false positive. And so the thing we would like to avoid is the false positive. Now, I don't know if this is true across fields. In our field, for sure, we have this culture that we don't make much of negative results. If we don't see a difference between the control and the treatment, we consider that there might be a difference, we just didn't do the right experiment, or the right experimental conditions, or at least in the lab, and I'm thinking genetics and cell biology, we tend to consider this as, and probably that's why so many positive results are published, or contributes to negative results not being published. It's because it's possibly in our culture considered to be inconclusive. Whereas when we see a difference, we consider that the difference is there, we see that as being conclusive. And so for the purpose of this talk, I'm going to conflate reproducibility with false positives. This might be something also that we might discuss. I'm going to come back to that in the end. But at this point, my worry is what I'm having in mind for the rest of the talk is having false positives. How often do we have? What is causing false positives? And then those are the results that somebody else is going to try to replicate and actually fails to replicate because it was a false positive to begin with. So I have the first question for you. You want to test an hypothesis. You have an experiment that you'd like to do. You're thinking of what experiment you'd like to do. So we're going to have either a possible positive result or negative result. And you would like that your positive result is consistent with the hypothesis you're testing, or the positive result, or would you rather plan an experiment where the positive result is not consistent with your alternative hypothesis, with your hypothesis that you're testing? Okay, so your positive result goes along with your hypothesis, or your positive result is against your hypothesis. You ready? So you're going to have 15 seconds. Don't forget to hit the submit button. "17 answers, and large majority thinks that the positive result should be consistent with the hypothesis. So there was Okay, so that brings me to the little I understand about positivism, which is a philosophical school of thought that indicates that empirical evidence interpreted through reason and logic leads to certain knowledge and truth. And this is associated with a Vienna circle in the beginning of the early century and uses the inductive reasoning. So for some reason, philosophers really like swans. And so the example that's always given is that if all the swans are white, all the swans we see are white, then we can establish that the truth universal is that swans are white. This has what is called the problem of induction, and that was, I don't know if solved is the right word, but a different framework as proposed by critical rationalism of Popper, which is the falsification principle, in which that we don't assume that a given fact is universal in truth. We can't possibly prove that something is true. We can only prove that it's false. And so in the example of the swans, we can't establish that the universal truth is that swans are white. It's actually when we find one swan that is not white, then we falsify the hypothesis. In the meanwhile, until that day arises, we just consider a conjecture that swans are white. We are working under that assumption, but that has not yet been disproved. And so supposedly modern science adopted Popper, apparently not so much in biology. So there was a controversy, very interesting controversy at the turn of the century, a series of letters exchanged on the journal Bioassays that got started by Robin Holiday, which is a very preeminent geneticist at the time, that claims that Popper is completely incompatible with biology. You can't possibly, so his argument is that you can't possibly disprove the genetic code or the Mendel laws of inheritance and that kind of argument. So I personally think that the arguments are plenty of fallacies, but there was a series of back and forth with scientists writing responses. For example, the first one, Jonathan Bard, actually disagreeing with Holiday and actually claiming, and with a very nice response, that Popper is important for biology and that we should embrace falsification. But actually, I have the impression that most biologists, whether they realize it or not, actually agree with Robin Holiday and are positivists. And actually, one particular quote of the Robin Holiday's argument is that a theory makes a prediction. To falsify a theory, the prediction must be shown to be wrong. To confirm it, the prediction must be correct. The actual experiment to falsify or confirm is one and the same. This is simply not true. If you want to confirm an hypothesis, you think of an experiment where the positive result is consistent with the hypothesis. If you want to disprove it, you think of a different experiment where the positive result falsifies the hypothesis. So this is, I would argue, positivist thinking. I think it's very widespread in biology. And one thing we should do is actually listen to Popper and think of experiments to falsify our hypotheses rather than confirm them. Confirming the hypothesis might just lead to false positives, if nothing else, because it's perfectly possible to have a framework that is false, an hypothesis that is false, an experiment that is designed within that framework is consistent with the framework, even though it is false. If you do an experiment to falsify it, then you realize that the framework is wrong. So I think we have a lot to gain using falsification rather than confirmation, confirmatory studies. Okay, so now for the rest of the talk, I'm going to be using this hypothetical example of a very interesting biological question. You found, maybe serendipitously, a super interesting novel structure in stem cells. It's in the cytoplasm. I don't know, imagine it's like a membranous organelle, like liquid phase separation, messenger RNAs. It's asymmetrically segregated to the one of the daughter cells that is going to be the stem cell upon the division. So this is really cool. And so you want to study this. Your first approach, prepare your smartphone. Your first approach is you decide to study this novel structure. The first question you should address is, one, is it involved in cancer? Two, what is the function of this structure? Three, is it important for stemness? Four, does it have a function? Okay, think of what your answer to the question would be. And I'm going to click here, and here we go. You can answer 1, 2, 3, or 4. Okay, 19 answers. Does it have a function? The majority of the answers. It's interesting, that would be my answer as well, but I don't believe you. I think most biologists, maybe it's because my students are here, they know what the expected answer is. Most biologists actually ask, what is the function? Rather than asking, does it have a function? And so then I would come to outcomes rather, which is the principle of parsimony, that we should always start. It doesn't mean, I don't think that the simplest explanation is necessarily true or has even more likelihood of being true, but for practical purposes, we should start with the simple questions and build on that. So first you show that the structure has a function, and if it does have a function, then you ask what is the function, and so on and so forth. I think we have plenty of examples of confusing the two questions or going straight at asking what is the function before actually it has been shown what the function is. We all know that outcomes like the simplest explanation, but the professor actually likes the complicated one. And actually, even more so, when we have big data, imagine transcriptomics or metabolomics or that kind of, we always imagine that the hits that are with a strong, with a high value, necessarily have a function, and have a function in whatever process you want to study. And it could be that it's just a consequence of whatever treatment you used, or it could be that it's completely spurious. And nothing here, this discourse of, I don't actually have an hypothesis. I let the data establish the hypothesis. Or that kind of reasoning is underlying, which I think it's wrong. We should have actually, first of all, clear idea of what hypothesis we're testing, what is the predictions of the hypothesis, and then look at the data. And that reminds me, one of my favorite quotes from Theodosis Dobzhansky. Scientists have often the naive faith that if only one could discover enough facts about a problem, these facts would somehow arrange themselves in a compelling true solution. So not only we don't necessarily go for the simplest explanation first, we actually have this intuitive, not carefully thought out approach to hypotheses and theoretical frameworks, that the data will actually give you the answer. This actually can lead, or talked about this, the harking or the Texas sharpshooting fallacy. The data will arrange themselves in a true and compelling theory, actually it's like your cognitive biases drawing the theory for you. So here I think the problem we have underlying problems of reproducibility is that in biology we don't spend enough time doing conceptual work. We don't have enough theory. So here and also we have this also comes to the problems of open data when you have this big omics. I'm not a big fan of omics, you might have noticed, but omics studies. We actually also need more open data so that those can be reanalyzed under this conceptual framework. But I would argue that we definitely need more theory, interacting with philosophers, and more time dedicated to thinking through our theoretical framework. And I'm just going to illustrate that with an example of a classical paper, the paper that reported the discovery of a messenger RNA by Brenner, Jacob, and Messelson. The first figure of the paper, it's actually laying out the theoretical framework, the different hypotheses that are being tested. And what is really important in this paper is that the first two hypotheses were refuted. And then the rest of it, we know, messenger RNA, we know what it does. This was actually in that field, if you go back to those old papers, common to start with the framework in the first figure, explaining the models, and then testing them. So coming back to our super interesting structure, the next question then you'd like to ask is, you have this structure, you are an expert in a specific pathway or gene that you've been working on for a few years. And would you go and investigate whether that pathway or gene or process that you are expert on is involved in this Sims function? Okay, so you're an expert in, I don't know, p53, EGR, EGF, all those things. And so answers will be yes, of course, two, it depends, no, third option, no, why would I do it? Okay, so if you know what you're going to answer, let's go to it depends, that's the most answers. Some say yes, some say no, why would I do that? Okay. Okay, so to illustrate what I think about that, I actually go to an old joke, and I found a cartoon from 1942. So Mutt and Jeff, so Mutt is on the ground looking for something, and the policeman comes and asks, what are you doing? He says, I'm looking for the quarter I dropped. And the policeman asks, did you drop it here? And he goes, no, I dropped it 2 blocks down the street. So then why are you looking for it here? Well, because the light is better here. So this is actually a well-known observational bias. It's the street light effect or drunkard search. Which means that you look for things not where they are supposed to be, but actually where it's easier to look for. And so in biology, we call that the candidate gene approach. And actually, Marcus mentioned that earlier in his talk. In human genomics, people passed beyond this and quite successfully. But I think in the lab in cell biology and cancer biology, we are still very much using the candidate approach. Which basically looks like that. You have this structure, you wonder if RAS pathway is involved, it isn't. So you wonder whether EGF is involved, it isn't. And then you wonder whether Notch is involved, and you get a positive result. And so that is great. You can ask for men, but the probability, if you keep testing these things on a biased manner, the probability that by random chance you have a positive result is actually quite high. And it's not only that you ask whether knowledge is involved in formation of the structures. Is it involved in the size of the structure? Is it involved in the number of the structures? Is it involved in the localization of the structures? You'll eventually find a positive result if you keep looking for it. And so this is probably has a high likelihood of being a false positive because it's a very biased way to search for things to begin with. And on top of that, not only has the reproducibility issues because it produces false positive, it actually hinders novelty. If you look for things that you are expecting to find, you're never going to find anything new. Of course, this is because of the incentives. It's cheaper and quicker to do for a candidate approach. But all genes that we now know the function of were actually sometime in the past unknown, and somebody found their function by doing unbiased approach, namely genetic screens, for example. So the candidate approach is biased, it produces false positives, it hinders novelty, and so I think the solution is basically just abandon that and go for unbiased approach. We have now pretty good tools to go for unbiased approaches compared to the old days. So it's longer, it might be more expensive, that's probably why it's not done so often, but it's probably for sure better. Okay, so now we found a mutant with a totally non-biased approach that may or may not be involved in the formation of these super interesting structures. So the question I wanted to ask, and we touched on this before, so I'm expecting everybody to have a 100% answer, is when you looking at your results of your experiment, do you know at all times what condition you're looking at? Control versus treatment, well, wild type versus mutant. When you're looking in a microscope, do you know what you're looking at, what condition you're looking at? So 3 possibilities of answer: yes, of course; no, but I should know; and third option, no, that's wrong. To know what you're looking at is wrong. It's the third option. Okay, so 1, 2, or 3. So this should not hide that until everybody answered. Okay, actually, no, that's wrong gets the most answers. And actually, we talked about it, but I'm going to mention the original, well, one old study that illustrates the point here, which is the study comparing bright rats and dull rats. So there were 2 strains of rats that have been bred over several generations to perform either very well or very poorly on this maze, where the rats would be placed in the maze. The light or dark sides of the maze would be randomly positioned, and so they would have to look for a reward, be able to choose where the reward was. And so students were given rats and were told to make the observation and see whether the rats were performing correctly or not. So suppose there's some genetic element underlying, because these rats have been bred for several generations and perform well. The idea underneath is that there's a genetic element. And so the students did find what was expected. So the bright rats had a higher rate of correct responses and in shorter times. And actually, even the students reported subjective perceptions, so that the dull rats were aggressive, the bright reds were clean and pleasant, and there was actually only one strain of rats. They were all the same. There was no genetic difference. They have not been bred to be performant or not. This was a study on behavioral, on behavioral psychology on the students. And actually what we could see is that the students found the result that was expected. So this is confirmation bias. The problem is that we see what we want to see. And there's a simple solution for that, which is blind analysis. So I always tell my students, implement blind analysis, but that's not very common in research labs, as far as I know. And it's actually, in some labs it is, but it's not on the materials and methods, which is actually something that should be standard. You should be doing blind analysis and you should be putting it in the materials and methods so that everybody knows that it's being done blindly, and actually it gets into the culture of the field. But definitely experiments should be done blind. Okay, and now we want to ask another question, which is, does this structure have an impact on cell cycle? Okay, so we are very fortunate because we have a drug that actually impairs these structures. The structures disappear. And so what we do is that we use this drug and then we analyze cell cycle in the stem cells treated with the drug, and then we compare that with the control. And so the question is, if we observe a difference in cell cycle, do you agree that we can conclude that sins are involved in cell cycle? Okay, so this is the question. If treating the stem cells with the drug affects the cell cycle, is it legitimate to conclude that sins is involved in cell cycle? Yes? No? I don't like the question. You can only choose one option in this one. It's either yes or no, or I don't like it. You can say no and I don't like the question. It's only one. So choose which one it is, and you can answer now. Don't forget to hit submit. The most answers we had so far. It's nice that the number of participants is increasing. And no, yeah, it's interesting that we don't have like a consensus on any of these questions. I would agree with no, by the way. I'm sad that some people don't like the question, but I think it's a nice question. So actually, I think this is a case that's quite common, I think, of correlation is not causation. It's like in statistics, in this case, it's in an experimental sense, but it's often not taking into account that the drug might be affecting the structure of interest and the cell cycle, but the cell cycle independently of this one, in which case you have a correlation between these two variables, but you don't necessarily have causation. What you know is that the drug is affecting the cell cycle. Whether it's going through this structure or not, you can't conclude. And actually, in classical genetics, this problem has been dealt with. So you know the solution. In classical genetics, we use epistasis. You can use double mutant combinations. And in this case, you could actually find a mutant or another treatment that actually restores the structures downstream of wherever the drug zeta acts, and see if that restores the initial cell cycle phenotype or not. So this is feasible. It's more complicated as an experiment, but it's feasible. It's a case of correlation is not causation. And then you can analyze the data. Okay, so the problem here is correlation is not causation. The solution is, well, defining properly your variables. Actually, is the way that independent variable is defined, it's wrong, or you can say confound. Okay, so now yet another experiment. We pursue this project on sins, and we have the drug that affects, that impairs the structure of interest. Remember the mutant XYZ, it actually encodes for this protein. So now we're going to analyze this protein on Western blot. And when we do Western blots, we need to normalize. So the question here is, in order to analyze the protein levels of XYZ1 on the Western blot, what would be the best reference for normalization? 1, GAPDH, 2, normalization, what's that? 3, actin, 4, total protein, 5, tubulin. Okay? So think of what your answer would be, and let's go. You can answer. Okay. Total protein, GAPDH, also you're fans of GAPDH. Yeah, normalization, you need to do normalization, otherwise you can't compute anything. Okay. And not so many actin or tubulin. So we need a normalization so that we have an indication of the total biomass we're looking at, so that we can compare things that are comparable. And just the number of cells is not enough, because you might be changing the number of cells. And so on Western blot, and this is a slide taken from my courses, there are plenty of steps where actually, when you go from the protein in the cell to actually the measurement of the signal, a lot of steps where things can go wrong, technically speaking, and where you can lose protein. Without going on to the details, if you have a difference in the abundance of a protein, this actually introduces a bias, because all these problems of saturation or overload on the gel or solubility might actually be different depending on the amount of protein. So you might actually be introducing a bias so that the treatment might be different from the control. And the thing is that nowadays the researchers say, oh, but you have these machines that measure the signal and we know whether it's saturated or not. Well, the signal in the end is saturated, but that doesn't tell you anything about all the previous steps where you could have problems. And so actually the standards are being relaxed compared to what used to be when people first started to use Western blots. And the thing is that there is literature on how to properly normalize protein. You can easily find that. Actually, when I looked for this paper, it took me literally 5 minutes to find a good paper on that. The best way to normalize is to consider results, total protein. If you are normalizing relative to only a single protein, this protein actually might change with treatment and control. So you are introducing a huge error if this is the protein that is changing and not that one. So that's a big problem. Another thing with Western blots in particular is that these papers tell you that if you have less than a two-fold difference, actually it might be purely technical and you shouldn't take that into account. It's probably not biological. You see in the literature increases of 50–60% of protein being treated as very meaningful. When we know that's not the case. So now this is not specifically for Western blot. Actually, I think pretty much any technique that requires a quantification of biological material is gonna be facing problems of normalization and technical errors. And so the problem here is that we actually trust too much in those instruments and techniques. We are really overconfident. And as things become routine, we actually become ever more confident and less worried about the technical errors that occur. So I think we should increase our standards on limits and so limits of the techniques and sources of errors, and on using proper normalization. So I messed up. I skipped the slide here, but fortunately I had just one slide to make the connection with Don's talk. About the questionable research practices. So this is just to remember what we just heard. And so the solution, we talked a lot about this just before. It's learn statistics and work with statisticians. And I would just like to show you this quote by Ronald Fisher. To consult a statistician after an experiment is finished is often merely to ask him to conduct a post-mortem examination. He can perhaps say what the experiment died of. So here the advice is work with statisticians and work with statisticians before you experiment. And that is actually exactly what Don just talked about before me. So now starting to wrap up, I took this figure from a commentary of Walden and Allen. I really like this imagery. And this comes back to something that actually Marcus already, well, I think everybody talked about this, that we can view research as a gradient, I would say, or a structure at different levels of rigor that we apply, going from pilot studies, where we're sometimes just looking for what are there as hypotheses that might be worth pursuing, and that we might have underpowered studies and not necessarily very clearly defined hypotheses, going all the way to confirmatory studies like clinical trials that are very heavy, testing one hypothesis, very costly, and this is fine. We can do pilot and exploratory studies. The things that we have to be clear, and again, this comes back to what Marcus said, we have to be clear about where we're at in this scheme. If it's a pilot, we have to be clear, especially with ourselves in our minds, that it is a pilot study, it is exploratory. So we can't really conclude much from it, other than what is worth pursuing with a more rigorous analysis. And what I actually really like is that this bottom part of the figure, that the traditional approach, it's quite blurry. The work we're doing in the labs and very fuzzy, whether it's exploratory or confirmatory. I think it is mostly exploratory, but we believe it's confirmatory. It's more like that. So we have to be, and pre-registrations probably help us with this. So, and I have the last question for you. When we are at this model of the experiments we do, imagine that our rate of true null hypothesis, this is just conceptual, this is coming back to Ioannidis paper 2005. The hypothesis we test, the null hypothesis is true 90% of the time. The alternative hypothesis is true 10% of the time. And we are accurate in our decisions 95% of the time. Okay, so the question is, what is the probability of having a false positive when you reject the null hypothesis? So you are here, and with this number, what is the probability of having a false positive? Okay, I try to be, I don't know, we have no idea whether these numbers are true. I hope it's fairly realistic, but so 5% false positives, 32% false positives, 50% false positives, false positives, I forgot to false positives, or 63% false positives. I made a mistake in there. It's false positives only. Okay, so 5, 32, 50, or 63%. There you have an idea. Go ahead. Okay, so most responses, 5% false positives. The list is 32. Okay, so I have to come back to the slideshow. So if I'm not mistaken in my calculations, this is the result, and actually you get 32% false positives. Okay, which under these assumptions, which means that even in a situation where you're accurate 95% of the time, and this was the whole point of Ioannis' paper in 2005, depending on this base rate, there's just a high likelihood of having false positives if the percentage of true null hypothesis to begin with is very high. So it's not just by probabilistic reasoning, it's not unexpected that you would have false positives. Now the thing is, how can we improve this and reduce the rate of false positives? So we don't know what this rate is. I wonder whether there can be some decent estimates of this rate by, I don't know, meta-analysis and curation of the literature. But it's definitely not trivial. I would be interested to see if that's possible. Definitely not trivial. But still, we could maybe increase the probability by being more stringent in what experiments we do. If you have a better rationale, if you have a more careful theory, maybe we could actually improve the rate of hypotheses being true that we are testing, and that would reduce the false positive. And we could also, maybe this is actually quite optimistic that we are 95% accurate in our testing. So here the problem is the base rate fallacy. So it might be an unpopular idea, but maybe doing fewer experiments, if we are more selective with what kind of experiments rather than the anything goes approach, would improve the rates. Maybe also a change in culture. We should be, don't trust anything until it's replicated. I don't know if this is a good idea. Please be always a bit skeptical, because knowing that false positives will always occur, you need probably a confirmatory studies. Now there's one paper that I would like to talk to you about. It's in experimental ecology. And basically, Yuku et al. They decided to do an assay on replication, but what they did was a different approach than what Tim did. Was they recruited several labs, give the same protocol to everybody. Everybody runs the same protocol in parallel, and then see if they can have the same results. So this is like several replications taking place in parallel, and then they're going to measure variables like the biomass of the plant, the root of the shoot. And actually, the first thing is that, well, maybe not so unexpectedly, very low replication. This is the effect size DC. So you can go from very null to a very strong effect, and sometimes effect in the opposite direction. And the other thing, and that stressed me out a little bit being a geneticist, is that when they introduced variability, and particularly genetic variability in their experimental system, they actually improved the reproducibility, which is completely counterintuitive to me. Well, it was at first, but within the same experiment, not across labs, just to be clear, within the same experiment, instead of using isogenic strains, they use strains with genetic variability. And then they come up with the hypothesis, maybe controversial hypothesis, that too much standardization actually leads to local truths. So you can have a true result within a certain lab doing the experiment under very stringent conditions, but then you can't replicate those results in other labs because of too much standardization. So it's proposed to as the standardization fallacy. And so when I talk to my students about this one, I try to view it as these are different experiments. And so you have all these are negative results that revolve around zero, and this would be noise. And then you have a very strong effect going to be a positive result. Whereas if you have high standardization, you might actually reduce the noise to a large extent, but actually might have a few ones there that you don't know whether those are real positives or false positives. And so maybe those might be increasing just because of high standardization. There are a couple of papers in preclinical studies in cancer, and not just in cancer, that kind of indicate in the same direction. At least introducing variability doesn't seem to hinder reproducibility. So definitely something to think about. I'm still on the fence on this one. I've been on the fence ever since I saw the paper for the first time. Can't make up my mind, but maybe standardization is something that it's hindering reproducibility. Now, just to finish, coming back to the question of what is a replication, how we define it. Actually, I think, of course, when you say replication, you should expect the same result, and this comes back to the question I asked Tim earlier on. There's always going to be variability in an experiment. You can't expect to have the same perfect, exact replication of the study. So if we, in my naive view, if we imagine the replication and we have like infinite replications of a given study, in this case, no effect, we could have, I don't know, maybe some kind of a Gaussian around zero. You do one first study and you have a positive result. And you believe it's a positive result, and then you have another one, a replication study, and you have the effect in the opposite direction. So in a sense, this is replicable. I mean, you have the replication of the results within a given framework. Given the variability, underlying variability of the framework, these are probably true results. And so I've been reading, I read a couple of preprints on this. I don't know, I don't understand everything, especially the statistical part of it seems very complicated. So I don't fully understand. But the point they make, and that I agree, is that we need theoretical foundation, actually quantitative modeling, probabilistic and statistical modeling of what we mean by reproducibility, so that we can make predictions of how reproducible our studies are going to be. So that because we don't know actually what is the expectation of how reproducible a given experiment is. What is the underlying variation? Unless we do 50 or 100 replications, we don't know how variable that's going to be. So we need a better theory, and they are working towards that. For now, they are working, what I understand, just with assumptions, not empirical data, but they are already defining parameters that could actually influence the replicability in a statistical sense, in a quantitative manner. So with a better theoretical framework for what we should expect in terms of replicability and all the other things we talked about, theory, pre-registration, open data, better statistics, and better technical methods, maybe we would expect more replicable, maybe lesser underlying variations so that we can conclude. The problem here is that when we do these kind of studies and we replicate, it comes back to the decision making, it is not very useful. We can't decide whether our hypothesis is true or not. So we need this better theoretical framework and improve the way we do the research, so maybe we can have better reproducibility. So the last quote from Saul Spigman, this is like 50 years ago when Nixon, President of the United States, decided to launch the war on cancer. He said, an all-out effort at this time to find a cure for cancer would be like trying to land a man on the moon without knowing Newton's law of gravity. We might have made some progress in the last 50 years in understanding cancer, but if we have reproducibility problems, we might have actually a bigger problem that we might believe we know the laws of gravity, and we don't. And that is, I think, the big problem with reproducibility, is that we might actually believe in things that are actually not long-term results. And thank you for your attention. Yeah, but
I can start.
Yeah.
No, this is, so you suggested in the beginning we should try to refute our hypotheses more, but then I found it interesting that you also suggest we should first look at a simple question like, does this structure have a function? Right?
Yeah.
And this is an existential question from a logical point of view that you cannot possibly refute. So that's also in Popper. He talks about universal statements. You cannot confirm them, you can only refute them. But existential statements, you cannot refute them, you can only confirm them. So to show that structure S has a function, if you show the function, then you've confirmed that question.
Yes. So if
you a positive response to the question.
If you're trying to make the point that my argument is contradictory, that's very likely.
No, but I was maybe
yeah, I think it's the existential part. My thinking is coming from a geneticist point of view. Not in an extent for us. Does it have a function means you have a mutant or a drug that disrupts that structure, and do you see a phenotype? Do you see like an alteration in some meaningful physiological, particularly we look at viability? Is the cell still alive? Is it still dividing? Is the embryo still okay in giving an adult animal? Some of you might have noticed this is actually inspired in a real literature example, which is the condensates, now they're called, which is well known for 20 years now that when you have mutants where those structures are missing, the cells are fine. At least in yeast and C. elegans, they're fine. So in that sense, they don't have a function. Now, is that an experiment that refutes the hypothesis in a Popper sense? I think actually I preach that we should use Popper. It's very difficult to come up with experiments to falsify hypotheses. It's much easier, much more intuitive. Probably that's why we do it, to have experiments that confirm them. Yeah. In the positivist sense, inducted, that are consistent with your hypothesis, where the positive result is consistent with your hypothesis. But that's why I showed the paper from Messelson, Jacob, and Brenner, because actually one thing that we can do that is have several hypotheses to be tested, where only one can survive. And so the other hypothesis, you have to find a way to falsify some of the hypotheses. So that would be, so to have several hypotheses being tested, some of them are going to be falsified.
Now, just if I may, just to clarify, I wasn't saying this is inconsistent with Popper to confirm an existential statement. So if you find that under some intervention the structure creates a phenotype, then that's perfectly fine to call this a confirmation, and that's not against Popper. But what I was saying, that trying to refute that is impossible. You cannot possibly test all possible interventions or contexts. So I was just asking you as a biologist, if in all experimental assays you've tried intervening on that structure doesn't create a phenotype, do you consider the structure doesn't have a function? I mean,
yeah, that's what we consider. And if we now whether that is falsification in the Popper sense, I have to think about it. I would argue that the mutants that don't have granules and are viable disproving the hypothesis that this structure is essential. So, Frederica.
Yeah, I would like to comment. So thanks for this presentation. I really like the street light thing. And this happens very often in our job. I've been studying a multigenic family of a protein that is involved in many processes in the cell. So I expected that if I would have all the sequences and make a nice phylogenetic tree and read all the functional annotations available in the literature, I would be able to say that this part of the sequence is involved in that process. Okay. But in fact, we failed because what we have in the literature, the annotation, it shows the subject on which the research team works. Okay, a research team working on a stress resistance will only produce annotation about stress. So if you want to understand a complex picture, you can't use the scientific literature because people are just looking for what they want to discover. So I was pretty disappointed. This is my street light problem.
Actually, it's even worse than I thought.
Yeah. Yeah, because we so.
But when I say unbiased approaches, I think we could use the protocol you described, but not considering the expected functions. Take it out of the picture, and when you do, if you do functional studies, if you look for a given phenotype, define a priori criteria that you consider like a positive hit. If a gene is involved in this, regardless of predicted function, if a gene is involved in this process, you should expect to have this result. And then if that comes up in your list, then you're going to study that gene. And if it's a novel gene that nobody studied, I think that should be viewed as great. The unknown function genes are probably the most interesting ones. Yet people go for the ones that have been like P53. Look at PubMed, how many P53 papers are out there. But we can use the kind of genomic approach you mentioned in an unbiased way, I think.
Thanks for that. I really enjoyed that. So I have a number of questions, but one I want to ask, and I just want to make sure I understand it so I can think about it more. The graph you showed of the Gaussian distribution, and it is a tough one, right? Which is like, did it replicate or not, right? So like, that's a really important one. You were centering it on 0, but I think what you were trying to say is like, there's a distribution of possible, likely, true effects, not that it's replication, even if it's on the opposites. I'm treating that 0 as if it's like a null. Are you trying to say like there's just a distribution of possible solutions, and that within, so that's essentially kind of like the confidence interval, right? Confidence interval or prediction interval can serve as that. It's a logical space of which you'd expect an effect to exist.
What I was thinking of, and this is my statistical limitations, is for one given experiment, you have infinite replicates. This would be the effect size. Okay. So here in the x-axis is the effect size. And the true result would be 0.
Okay, perfect.
And you do one experiment, and you might end up, and maybe I didn't mention this. If this were true, you can have results in this anywhere in this distribution. You could expect to have the replication actually increasing the effect size, not decreasing. And you show that's not the case. So obviously there are other issues like publication biases and whatnot. But what I was thinking is effect size, you do one experiment, you get this, you do the replicate, you get that, but that's within the possible distribution of that experiment.
Perfect, that makes sense. Yeah, I was just treating the 0 as like 0. I would have used a different number. So that's what threw me off. Yeah, so this makes a lot of sense. And I think there's something very interesting with this aspect of it. And it's actually great that you drew replication kind of on the smaller side, suggesting you possibly even have a smaller effect, as there's this constant like reduction of the mean, right? Like we see a decline effect. And that's like really notorious. And this is basically, I think, like there's a part that I struggle with a lot here, and Marcus talked about it. I mean, we've kind of all talked about it, which is the way we go about conducting and communicating research. We're setting ourselves up to keep making it look this way, right? We keep doing small sample studies. We keep trimming our data. We keep making completely inflated effect sizes that sit always that far, even though you're right in that true, theoretical graph, which I wish we knew. If we did, we would have our replications tend to always go down. So I'm just wondering, like, what do you make sense of that, right? Like the fact that like we just keep doing this practice, and we've been doing it for a while, where we just keep doing small studies and we keep pushing and kind of presenting things to make them look so nice, and that even if a replication is a success when it's lower, like we just don't know, right? We don't have infinite resources to explore this whole space. That makes sense?
Yeah. I think it's the issue of when we talk about publication bias. I think it's the whole process up until publication. Even like when you're doing exploratory studies, if you have a small effect size, you're probably not going to pursue that. So I think in all the steps of the process, we are biasing, and it's because we are looking. It comes back to my first slide. We are looking for positive results because we have meaning difference between control and treatment. Because I think in our field it's very entrenched that the negative result, the low effect, low magnitude of the effect, it's not meaningful, and so only when you have in fact it's worth pursuing.
Yeah. I think just as a note on that, we can definitely talk more later. There's a cool, what I'm trying to get at, there's this really cool play you can do on meta-analyses, blanking on the name. Basically, it's like a Christmas tree. And you can see publication bias on it because of what you're just saying, right? Like we see all these positive effects, they sit there, and then we have like half the tree is empty, even though we know it should exist, right? Like we don't publish like your left half of, you have a tree there essentially. We don't publish what's on the left. We only publish what's on the right. And so we have this huge distortion, which is really frustrating because on one level, that's the distribution of what we have, and statistics help us, and the p-values are useful, but they're actually not what we care about, right? We actually care about the fact that it's above 0. And I think that's the part that I really struggle with, which is like, how do we wrestle with the fact that we keep tricking ourselves and wanting to publish things because we want them to be biologically significant. And yet we have this huge Christmas tree problem, which is half the trees lit all the time.
I mean, I think it's inevitable. Marcus also said we're humans, so we have to, and so that it's inevitable, and so we should embrace uncertainty, which is, by the way, also a good reason to embrace Popper. That at least it's always conjectures. You never view it as definitive. And so definitely, I think the culture of uncertainty is also a good way to deal with it. I mean, we are going to publish false positives for sure. And actually, 0% false positives in the literature probably means that we're not taking enough risks, we're not looking enough novelty, we're not moving the field forward. But I think it's more the problem of having too much faith on published results. So we have to deal with uncertainty and accept uncertainty, because we are always going to, like you said, look for the effects on the right side of the curve. No matter what. And probably there, I mean, there's some, I think it's logic to it that if you're doing exploratory results and you have no effect, it's probably, there's nothing there. And so you're probably going to a dead end anyway. It's probably your understanding of the phenomenon is wrong, and it's really not worth pursuing. And there's always this, it could be random, you just have bad luck, or it could be that it's not the right question to ask, and so you have a negative result, you move on to something else. Maya?
Just a question on this graph again. So follow-up question on the meaning of the graph. So I mean, the curve, what the curve represents is that the middle curve is more like the most frequent result you would get from in an infinite series of the same experiments.
Yes.
Okay.
So here would be the frequency.
Okay. So then in that case, you don't need to say, I think you said that this point corresponds to no effect size, like null effect, or so this is what you said, right? But this should be dissociated. These are two different things.
Yeah.
And so you can represent the most frequent result you would get in an infinite series, but you can also say this is where we have an effect. It's a different point on the line, right?
Yeah, you're absolutely right. And actually, that does come back to what you were saying. It's a mistake to put a zero on it. So I wanted to use as an example one given experiment, hypothetical experiment, where you have no effect. But I don't need, you're right, I don't need to give that example. I just, I wanted to do that so that this would be a false positive. But the idea I wanted to get at is that results are going to be inherently variable. So when you do replication, it's going to be within a certain range of variability. And so the thing is that how big is that range? And if you have a range that is so large that you can get results that are clearly a strong effect in one sense, or results that are in the opposite direction, when it comes to making decisions, this is not useful. So when you get this replication, what do I do with this? What do I do with my hypothesis? And so we need a better theoretical quantitative model framework so that we make those decisions. But I wanted to use as an example, null hypothesis is true. It's what I wanted to use as an example. Maybe it wasn't clear enough. Yeah.
Thank you. Yeah, thanks. That's really interesting talk with lots to think about. I really liked when you went back to and Edith sort of talking about, given that you do get a positive result, the chance it's actually a true positive. And I think that's, even if we were doing the best work and there wasn't a publication bias, that would be true. That's the truth. And of course, the best scenario would be, I think in this table he had clinical trials where the evidence is equivocal. So you think it's a 50-50 chance that it's true, or H0 is true or false. And so even then, it's much worse than 5% if you're So it's helpful. I think it was what was illustrated of that table in his paper is that actually sort of it almost explains why we have this reproducibility crisis, because those much more speculative research, because of the publication bias, are much more likely to then get published, and hence that was the alarming numbers that you put there. But to some extent, if we recognize that and have more, so say you would decide what you looked like to me, what you were designing before, when it's does it have a function? You were proposing, and I probably haven't fully understood what the context was, but it sounded to me that you were going to look at some sort of array where you could look at everything that you know about, maybe, and test on this in this particular condition to see how many of these measurements achieved a certain threshold. I
think we had so that thing with the structure. It's a cognitive bias, I think. Not only for biologists, but we will have the cognitive bias. But it reveals in biology that when you see a structure, you assume it's doing something meaningful. Where you're going to look at a bunch of variables, sometimes biologists have very specific ideas. What is the things that are being looked at that are going to be altered? But there are a lot of structures that actually don't have a function. But we have this cognitive bias, and then it's very trendy. Actually, these structures that are very conserved and we find it in different organisms in different contexts can get you very good publications. even though it's been 20 years and we still don't know what you're doing. So it goes to it's with the publication bias.
But I think it would be possible to approach the experiments that you do in the discovery space through to confirmatory with frameworks that do, at least you have like a pipeline. Maybe we could think about sensible frameworks for discovery phase experiment, recognizing that you're going to be looking at lots and lots and lots and lots of things. And then they only get carried forward to the next stage if they meet that threshold. Because ideally, you would like to be in control of having a good guess of what these priors are, because the prior data, it could be there in the literature somewhere for us to know it.
Yeah. Well, that's actually a good point. Actually, one thing might be related, but I think there's a lot of examples in the literature, the things that are replicated, and we don't know yet because we don't have a system of curating replication in the literature. I think, I don't know if you agree with this. Maybe we can move to the discussion part. I have the impression that often somebody's result is somebody else's control. And so they pick a result on the paper, they build on it, and they use the previous result as control in their experiments, and then they add one more variable. And I don't think there's this curation of this kind of replication out there, and that I think would be useful. We have a peer getting a grade for a number of things, but similar kind of things. And people in the field might know the literature enough, like a very specific field, might know the literature that know that this actually has been observed in 3 different papers. So we are pretty confident. But people from outside the field don't know all those papers. And I think those, I might be wrong, but I don't think those are curated unless you know like very well your literature in a very specific field. There's probably a lot of replication out there that is not accounted for. Kind of an optimistic note. Yeah, but I see we do have to talk a lot more statisticians and try to get you think about these experiments. This was really, as I think about it as a biologist, but so I want to be statistics and then what kind of statistics studies you do are probably not clear in my head.
Well, we could maybe start with one of the questions you raised, I guess. What is replication? That one. Yeah, I mean, many people may have their definition, but it doesn't matter what word we choose, but the arguments are interesting. So because it will emphasize what you think is important in this situation. I'm proposing that we start with that question. How we should call that crisis, but rather what is the most important thing? Why do you, for instance, Tim, you have done that in the paper, you have tried to define replicability, replication, so that might be a good starting point. I don't know, what do you think?
Can you start with a simple, with an easy question?
Yeah, what is replication? You prefer, what is replication? Yeah.
So I'd like to have the first go at this. So I think it's important that you make it clear what the context is. So you've got, as I said before, the statistical error rates control all of our results. Whereas what really is with this is this answer really true? And I think that's really, it's going to sound wishy-washy, but it depends how much evidence you need and what you're going to do with that result. So, for example, in clinical trials to have shown statistically significant result before they will. So they make that clear. They may well be missing some treatments because of that. They want that much evidence. Whereas you may, it depends what the consequences are. Are you going to then a whole load of
what you want to do with the evidence, what you commit to with the evidence. Yes.
But when you're looking at, when we're trying to look at research culture, then we would expect, if we were in a perfect world, results that have previously been shown to be true, and we would expect really high rates of replicability. But only looking as a whole, you know, of 100 studies that were repeated, we can have a definition of replication that works for that, thinking about labs in general and experiments in general. When it comes down to a specific question, maybe more about how much evidence do you really need to be confident that this is.
So it's defined, I mean, in the context, for instance, of clinical trials, this is defined pretty standardly. Of course, there might be disagreement on where we should put the bar, but or the threshold. But in preclinical research, for instance, what would be, I mean, how could we quantify when we think that we have enough evidence to trust a result? How should we, or how could we do that? I mean, we could say intuitively, we could say it's maybe less important than in clinical trials because it's not the lives that are at stake. It's more like research programs, the directions where scientists would go, where funding bodies would invest their money, and that kind of questions, which are of course important in the long run. But can we quantify that?
I don't think so. So like, I don't think we're ready to. So, and the question, right, being of like, what counts as if it's replicable? I think I agree with you. You have to state it. It's what you want it to be. And I think it's back down to what that next direction is. So I, as an individual researcher, it is what evidence does it have to be sufficient to warrant further investigation? I, as a funder, is what evidence should it be to invest further resources to push that? I, as a practitioner, as a clinician, maybe if I'm taking it, is why listen to this? It's great you did it in animals, like, is it sufficient? Which is, to me, like, effect sizes come up a lot because I think that's what I care a lot about, and eventually that has to translate. But I don't know if that's always the answer, right? Like sometimes it might be, oh, it's sufficient at this stage to see anything, a effect, right? A function, a structure, versus if you get farther in, then you might be more concerned about the precision of it. So I think it's a little bit, to me, it's always been about, which we didn't do on purpose, right? We actually completely punted on that, which was, we never defined it a priori. We were just like, we don't know the answer. So we're just going to keep grabbing all these different methods and seeing what we can do. And actually that's what those prediction markets were doing that Marcus showed. Just another way to guess at this, which is like, how do you figure this really hard nut to crack? And to me, I want to quickly throw out as a last thought, which is, it's not even about replicability, it's about confidence. How confident am I in what this claim is? There's so many factors at play. So I don't know. I personally think that like it's unsatisfying, but it's a little bit of like the eye of the beholder of like, what do you want? What are you hunting for? And so I actually think that there's high disagreement, and that's what we should maybe focus on, which is, if we ask this whole room, do you think this replicated or not? And we have more consensus that would say yes. If we have high variability, then we have to say, well, what's going on? Right? Why can't we all agree? Why are our points of view so divergent that we don't think this evidence supports? That's big, maybe. I don't know how you do that though.
So maybe a further proposal to just follow up on that. I mean, there's this paper that is very famous in philosophy of science that has been done on research in general. So it's a very abstract paper. And the idea is to represent research as a field where you have hotspots that are places where you can make important discoveries. And the idea is to model behaviors of researchers. And you have two types of behaviors. First one is very conventional. That is, you investigate the area where everyone else is investigating. So it means that you have a high chance of finding something, but not a high chance of finding something very significant. And the other type of behavior is the behavior of the mavericks. So the mavericks would precisely go where the others are not going. The idea is to try and do simulation to see what is the best optimization of these two behaviors to find the hotspots more quickly. And the answer happens to be majority of followers and minority of mavericks. Okay, so what I'm thinking here is that what we are adding to that would be something like, maybe the decision should not be too systematic, like I'm systematically doing or looking in another direction, if I'm a maverick, for instance, or I'm systematically looking into the same direction as the others if I'm a follower, but more something like, depending on how I trust the evidence, I would maybe deviate a little bit or deviate a lot or not deviate at all or something like that. I don't know.
Well, one of those groups that the preprints I mentioned in the end that they are trying to make a quantitative statistical model. It's based on assumptions, theoretical assumptions of what reproducibility is. That's one of the parameters they use. Mavericks versus followers, and it does have an impact on their model of reproducibility. And that's kind of, they probably have read the same literature because it's a very similar conclusion. You need a fraction of mavericks, but not too many. And then you have like good reproducibility, but they also have a parameter of, I can't remember, something that indicates the novelty or the field moving forward as well. But that's one of the parameters they use, and they does need some mavericks in there to keep things balanced.
Let's maybe open the discussion more broadly to other participants. Frédéric?
I was wondering, but this is a question to also everyone, like what is the importance of skill in experiments and maybe like tacit knowledge that there's only so many things that you can explicitly transfer to someone when they're trying to replicate your experiment. I don't know, also if you're thinking of a cooking recipe, like, I mean, you've tried that maybe 20 times and you write it down that someone else will not be able to replicate it. So are there things like that are just, hidden in the tacit knowledge of the experimenters? And should we worry about that?
So I'll comment first. So in psychology, actually, this was explored in that psychology project. This was raised of like, and not quite the same way you're doing it, but it was like a criticism. Oh, you guys aren't experts, you replicators who did this. Only us original authors are experts, right? So there is this like inherent knowledge. So they did one of the studies and they had, they basically changed up where they had more input from those experts to weigh in on the design a lot more heavily than they did in the original replication project versus just repeating it kind of maverick style, I guess, on your own. They saw no difference. So they kind of like put that aside. It's not obviously definitive, but it's just like, okay, well, you made a statement that you guys are experts and you need to weigh in more. You felt you didn't weigh in enough. Here, try again. We'll do this whole experiment. It makes no difference. But at the same note, and I'd be curious to hear about like this from the rest of you, is like, I know from biology and like being in the lab, like there's a lot of like kind of silent knowledge that gets passed. And I actually think that's a huge problem because I think, not like in a bad way, it's an opportunity, right? Which is we do things and we communicate with each other, probably not even in written or verbal ways, and we're not communicating these essential aspects. And so this gets back to like how we train and how we then train that to get information out to be more equitable and disseminatable to other people. Because I think sometimes it takes somebody watching you to see what's actually happening. And I think what we all want to do is hope that it's not important, right? It's like, I hope it wasn't because I was standing on my left foot more, right? Or I pipetted slightly different today. Like, I hope that's not the problem, but it could be. And I think that's the whole thing of like the devil's in the details. And I think there is this tension probably that goes, especially with like a lot of more complex biology experiments, where there could be these hidden factors that are at play. We don't actually know about them, but we actually do need to start to surface them, which is why that's the stance I take a bit more, which is I believe when you can't replicate something, it's probably because they're both true and we just don't know what's going on, right? They're both happening and there's some tacit knowledge there that just never got transferred.
Yeah, I want to weigh in as well. I have very mixed feelings about that question. Actually, it's funny that you mentioned cooking, because one of the best things I saw on the internet, there's some fun stuff on the internet, was a cook trying to explain reproducibility of a recipe that was like a declination of a pois Belle. And actually, well, he was actually not for standardization at all. He was like really for adapting your protocol to the circumstances, what was his point. Because sometimes you had like a commiss power, and then the other day was like a different type of pair. And get so you have to adapt to that circumstances. And so I think if you do that in science, you're not going to get very far. But it's an interesting way to think about it. Actually, you want to get the final result, so you have to fiddle with the protocols to get the final result. But then if you think about cognitive biases, and if you start fiddling with the experiment to get the desired result, then in science, that's going to be a big problem. Another thought that I have is that if these experiments are really contingent on somebody having a skill, and somebody else can't reduce the experiment, how robust is that as a discovery? You know, I also have the same issue with the replicability. If you have a given result using HT16 cells, but you change your cell type and it's not working anymore, then it's true for that first set of experiments. You can't generalize that, and it's in vitro. So you want to know what's happening in cancer in vivo. So how relevant is that? So if the experiments are contingent on skill of a given person, they might not be very robust. The counter example of that is the paper I just showed you of the discovery of messenger RNA. Was Sidney Brenner, who was both brilliant intellectually and practically, excellent experimenter. He found that they needed like ribosomes to translate messenger RNA in vitro. There was one little detail that nobody could figure out. Magnesium in the solution. It was Sidney Brenner who solved it. I mean, so it does depend on skills of people. And in this case, it was very important discovery, and you need to have the skills. And actually, it's same paper, they had to go to California to Masselson's lab because he was the one who could do radioactivity. So you have twice the example of that in that paper. But those are things that in the end, you can put on a protocol and other people can reproduce. So, yeah.
I mean, I suppose just to add another layer of answer, I'd say that there are examples I'm thinking of where in a lab, for instance, I remember a colleague of mine who was the most gifted to slice the brain of a mouse. And I don't think this is biased toward any result. I mean, you would get the best picture from that. So whatever the hypothesis you would be making, you would ask him instead of anyone else. So, and that is improving the quality. It's not, I mean, this is definitely something that is, yeah, a part of reproducibility, but not a part that is biased toward any result, I would say.
Yeah, I wanted to say a similar thing. So it's not fiddling to get the result you want. It's fiddling to get the best conditions for seeing an effect, if it's there, right?
So
I think that's
of this talk mentioned a very important aspect of that, which is we first got the controls to work.
Yeah.
So if you get the controls to work.
Yeah, and I was gonna say like fiddling. Yeah, that's a dangerous word, right? Like fiddling. But that optimization. I'll use that word because I feel safer. To me, the thing that's tricky, and this gets back to like theory and model development, right? Which is it's okay to do it if you know what you're trying to look for, right? Like if you're doing cooking and you're saying, listen, I need to adapt the oven temperature because if you're in a different elevation, it's going to cook at a different time. I'm not telling you that you have to hold the oven temperature constant. I'm telling you actually the more true statement, which is the temperature has to be relative to the environment that you're in to cook internally at a specific temperature. So like when we start to develop our theories better and our models and our understanding, that's when I feel like optimization works the right way, which is I'm not telling you what the result is. I'm telling you the proper conditions. And I'm just using, because I don't know it, which is I'm just going to tell you how fast I spin it. When really maybe I should tell you RPMs, or maybe I should tell you something else, which is actually the gravity that you're pulling. Like that's the way I should talk about the way that I spin my tubes, for example. But we forget all that, I think, as we move through. So to me, like I like that. As we mature, you start to see that optimization actually move the right direction, which is it's not actually like per se the mechanical things you're doing. It's the theory behind it that holds it constant. That's a good example.
Other thoughts? From people in the back row or back rows?
Questions?
ask a question in the back row?
Of course you can.
Okay, so you heard a lot. I'm trying to put myself back in your shoes because I actually really obviously enjoy this stuff because this is what I get to do now. And I'm curious of like everything you've heard. I can't imagine. I imagine if I were you, I'd be like, you guys just told us a whole bunch of stuff because we just did tell you a whole bunch of stuff. What's working and what's not working, and probably a little bit on the what's not working. But like, what did you take away from today that's an actual practical thing that you can do? Like what, like, is there one thing that you can say, I'm going to do something slightly different the next time I go to the lab, or the next time I go back to my lab group, or the next time I read a paper or whatever that today taught you or inspired you? Hopefully something.
One more statistics.
That's a great answer.
About the data and the metadata. That's something, but actually, I started that the work being sure that the variability is in good and having two different results is like confusing. Now, as we end the day, I like more like it's good to have more cell lines and more variability. And if I can find the same results in different cell lines, even if they have different backgrounds, it's even better. And actually, I'm a little bit confused. I'm still not understanding how we could actually improve reproducibility in our labs. Because before the lunch break, I was like, yeah, we need more strict protocols, we defined methods. And now I'm like, yeah, everyone doing his source and it's working. So it's better. Maybe this way it's better. So I'm confused. I'm a PhD student in the second year.
Actually, I would say similar. So it's a little bit frustrating that it's so hard to define what's reproducibility. And yeah, so since we want to have same results, but if it's depends on the way how we mix our tube, it shouldn't be, it's very weird. And so I think that's the case why we are sitting here, isn't it? Because that's a problem to define it or to find a balance between these two parts to take or to do it always the same way and to do it in a different way to still obtain a similar result. And I think I will take, or in the future, I have it in my mind that this is the problem and we have to think about it. And yeah, just have it in mind that there is so much to think what's the balance or what could be the better solution, or not the better solution, but yeah, just have it in mind.
Yeah, I would add that although I'm also confused about this. So, but what I am not confused about is the one solution of the open science, and I'm looking towards looking to it as soon as possible, because it's something very interesting.
Other reactions? I have a question about this idea of increasing the amount of pre-registered protocols, pre-registered, I don't know, experimental design plans, which obviously it is very good for statistical significance. And it directly leads to this continuum from very exploratory research where we probably don't need this, and very, statistically loaded confirmatory research where we obviously need this. And I'm interested how it is practically implementable, because I'm not an expert, but maybe the, I don't know, professional biologists know better, that usually there is a big diversity of exploratory confirmatory statements inside one article, and maybe inside, I don't know, maybe inside even one experiment if it involves some complex procedure. Is it even feasible to always clearly distinguish, or it's we will always fall down to to just making servers?
I'll give you my perspective on that. That's a great question. You kind of raised it before. One is when you can, and it's mature enough to tease things apart, it's always safer to get them a little bit down into units. But this exploratory confirmatory continuum is actually a lot easier to think through if we think of it as a little bit like less threatening, right? If you're exploratory and all you have is a statement, just a general question, and you're saying all I'm doing is exploring, I'm just collecting data. I'm going in eyes wide open with what I'm doing. You can still pre-register that, like Mark was saying. There is a camp that would say that's totally fine. That action is just saying that and saying up front, listen, I went into this with no intention of doing it. In fact, you could also push your statistics to the side if you really wanted to, because you're just going to use exploratory. You're not going to worry about like trying to like look at a p-value because you're saying that wasn't my intention. You have to be honest with yourself. That's like a really honest question to ask yourself. And as you start to move down that pipeline, as you start to really want to, I think, tease apart and like formulate and, you know, hopefully falsify some potential models that are out there, that is when I think it all comes back to you really trying to hold accountable to how am I going to know if I'm actually able to do that, right? This is when I think statistics really play a high role. Pre-registration plays a really high role in terms of what's my sample size? How do I choose that? What's the specific question or questions that I'm asking? So I think there's a bit of a continuum that you're getting at that's true, that it's not like a one size fits all, and that it does have to evolve over time. And this is the last thought on that, and this is true in the clinical space too, which is you can update. If you're doing a pre-registration, like it shouldn't hold us to the point where we feel that we can't still explore. It's trying to hold us accountable to what we said. So if I, in the beginning, have a thought and I lay out a lot of details and I begin doing my experiment and I'm like, that's wrong, totally got it wrong. The formats that are available, the tools that are available, just like in clinical trials, is you can update that. The trick is that you're versioning it over time, right? It's just like data. You're versioning your data over time. You're just basically holding yourself accountable to, I'm making these changes and this is why I'm making those changes. And then hopefully, ideally, you get to the point where you can actually share that to the rest of the community. So that way I, as a reader or reviewer, can say, oh, cool, that's the journey you went on. So in the end, when you write your paper, I know that you changed things over time and that's okay. But you rationalize them at that moment in time, if that makes sense. Qualitative research struggles with this a lot because a lot of qualitative methods are by nature, like they have to adapt. And pre-registration is thought to be in conflict with it. I think there's a group of people that don't believe that it is.
Yeah, I would say basically the same thing, but when you're doing the experiments, exploratory pilot studies, you can still write down on your notebook. You have electronic notebooks. I think, I'm not sure if it's the Center of Open Science, but there have been tools developed that you actually have websites where you can easily just register before you do the experiment, while you're doing that. And actually this is the Dobzhansky that I showed the quote before. Ironically, he has a contradictory example to what he said, that the most important discovery he made in his life, he actually doesn't have any record of having written the hypothesis he was testing. And he wrote in the paper later on, I don't know why I did that experiment. I couldn't find any trace in my notebook, but the results were staggering. It was like some chromosomal rearrangements in the neo-Darwinian evolution. I can't remember the details. But even if you like kind of absent-minded like that, when you do an experiment, you do it for a reason. I mean, you don't just like randomly throw stuff at the microscope and see what happens. You have some kind of, however exploratory that is, you have some kind of rationale. So there's a reason why you did the experiment. That should be written down. It's my point. Just write it down so that you have a trace, because you might actually forget it. So that would be covered.
Yeah, I think you can. There's no harm writing down what you're planning to do, and it can be pretty concise rather than being too deep. I wasn't trying to frighten people with, 80-page statistical analysis plan. It's just a single page and just basically saying the main reason I'm doing this experiment is to answer this question. This is the outcome. This is how I'm going to analyze it. It can be very, very simple. Ideally, you need to talk it through. with others. And of course, you're not just doing one big experiment all your lives. You're going to be, you're either doing your PhD where you've got your own program, or you're part of a bigger team, you've got a program. So you need to understand what it is that you're doing, how that fits in to everything else, so that it more connects. And sort of writing it down clearly, because it just helps everyone understand that everyone's understood how this is going to slot in, and depending on what result we get, that will dictate the next stage.
Yeah, I see it as a tool. I think hopefully you'll see blinding as a good tool. It sounds like you guys hear a lot of that. Blinding is a great tool. Randomization is a great tool. Registration is a tool. These are tools actually to protect, from my perspective, they're to protect us. We're human. We're prone to these things. We see signals, we see patterns really easily, so we have to protect ourselves from it. Doesn't mean that you're doing anything like wrong. It's just to help you. It's really to help your future self. But I wanted to add one comment. I'll give it my perspective. Actually, it's to what was said over here, and I'm sure there's other voices over here that have it. But it's back to this question of like standardization versus full, like, heterogeneity of what we're doing. And it's actually it's an interesting topic, because I think that there's still a lot of split in terms of like how it can help. So I actually think that's a really fascinating conversation. I'll give my perspective just quick on it, just for what it's worth. I think I'm a bigger fan of transparency helping across the board, right? Like, but at the same note, I am a fan of making sure that we can move towards standard operating principles, because there's high value in that, so that way we can communicate and make sense of the data across each other. A lot of the variability that shows increasing in reproducibility tends to hang around a little bit more on the sampling size, right? So like genetic variability, population variability, like that's where most of the conversation lingers. So it's like, oh, right, we want to make sure that we have multi-centers doing this, because we want to make sure that there's not some variability of the way that the mice are handled, or the way that the mice are housed, or whatever that, which is really important, right? Because like, geez, we wouldn't want to have our experiment all because of the bedding the mice were raised in. We really are trying to guard against that. But at the same note, I think we do know that we have to do some of our experiments in a very standardized way in order to get those results to work, right? Western blots, as you just showed in a very simplistic way, you can't change that much. Otherwise, you're not doing a Western blot anymore. So I think there's this like weird tension that has to occur, which is we need to communicate our results, how we do them standard, and list the details, not because they matter, but because they might matter. Yeah, to me, bring in variability, scaling it up over time, because we don't want it to be about the many things that I'm probably just doing myself over and over and over again, because we're repetition, right? We're doing repetition in our experiments. And so randomization and variability is meant to guard against that, which is create a little bit more noise, because you don't want it to be that one thing that you're doing. So there's this weird tension, which I think most of you guys were calling out. And to me, it's the right tension to have, which is how do I balance the two? And to me, transparency is the one that kind of helps expose it. What am I doing constantly that I can monitor? Can't check everything. It's not like you guys are like checking the room temperature or the humidity every day, even though that could make a factor. You're not going to do it. And then where do I introduce it where I'm like, pretty sure I know these are going to be factors like the cell lines. I know they're going to be different. So why don't I just start introducing it on purpose to see if I can get a more robust result?
Yes, on the standardization fallacy, it's a fascinating topic in itself. I'm still on the fence. I don't know what to think of it. I think it's probably going to boil down, and I think we did a lot of work in thinking about that. Being able to wisely choose where you're going to introduce variability. Genetic variability is probably not this candidate, but I think there might be other things in the protocol that you might want to make variable, and others that you might want to standardize. And I think it's going to be way more complex than just introduce variability or standardize. It's probably different steps of the protocol. You might be super strict with standardization, and other ones you might actually want to have biological variation in order to be meaningful. Just to make things easier.
One question I have for everyone. There is a situation that is very well known in medical fields, which is that the first studies you find about the effect of a given drug, any given drug, are generally much bigger than, I mean, the reported effects are generally much bigger. And then with time, you can see that the reported effect is decreasing more or less quickly. Well, there is this tendency, and there, of course, there are exceptions. Is it because this is a field where it so happens that we have a lot of replication in various circumstances because you want to use, for instance, these drugs for test, I mean, like test cases against control, sorry, cases for about other drugs, or you want to check whether they work on another population and yet another one, and there are a lot of replication studies, so to speak. Therefore, we tend to see something that we don't see that much in basic science because we don't see the point of replicating, or do you think it's generalizable that we have this tendency to find a, let's say, a smaller and smaller effect with time? Because I mean, for me, it's very much in line with what I take to be the major result of the eLife paper. Like what is striking is that it's not so much, I mean, it's, you can find, in many cases, you can find the same effect, but the size of the effect is much smaller. So, I mean, for me, it's the same thing. Like it's, you try to optimize in some ways the results you get because obviously it's more convincing for publication. I think it's, to me, it's the same tendency, but I'm not sure.
PhD students, I tend to read articles. If I find an interesting result on the cell line, I tend to put positive controls and negative controls as my controls. And sometimes I will even use the cell line they use as my positive control. So there is a lot of replication in biology. I think that there is really a lot of replication. And if we can't replicate a result, be published, I think it's difficult. First, we ask ourselves what we are doing, and protocols are not clear. But a lot of protocols are usually used. They are published. There is even videos of the protocols, and the results are questionable. So I think this is why there is a decrease of the value of the findings, maybe.
In the drug trials situation, it's quite accepted that you've often got a regression to the mean feature, and a lot of that is, so you'll have patients initially seem to have a response which diminishes, and that is often because there's a selection built in the eligibility criteria. Someone already has to be quite severely affected, or not, they're not representative of the population. They're already in this, you know, in a special group, and that's why in those trials you see that also in the placebo group. So, or the, you know, some, so that is a well-known feature, which is why placebos are so essential. But I think where you tend to see this, the winner's curse situation, I feel quite persuaded it's a combination of the underpowered studies and the publication bias, and that you're just, if you've got an underpowered study, you're more likely to get a false positive, which has got a high effect size. It's all part of that problem. And then if someone else comes along with a larger study,
so the regression to the mean effect would just come from the fact that you have a larger and larger population where you have tested the drug. So you're closing in on the true value of the, say, the true size of the effect, something like that.
It's more that the people normally would just vary, you know, in their condition might just vary. And if you're just going to select people when they happen to be at a certain part of the distribution, then it's, some of them would just go down, because you're picking, if you imagine you're picking them at like when they've got high levels, when normally they would really fluctuate like this anyway. So that's why you can get that. And it doesn't mean that the treatment's not working, but it's just that's why it's really critically important to have placebo. And that's sometimes argued why placebos sometimes look like they're being effective.
Mhm.
They're capturing the regression to the mean.
Okay. Other questions? We have planned for also conclusion. I don't know when you want that to come. José, I'm looking at you because you're organizer in chief.
Yeah, I think, the conclusion is that we have a coffee and goodies at the end.
That's a very good conclusion.
I don't have very much to conclude. I think it was, I'm very happy with the content of this presentation and the discussion. It was great. I mean, it was a great success. I think about the expectations we had. They were completely met. I'm very happy to see the students here. I'm happy to ask the question what they took from it, because I think that's the important aspect. Yeah, this is just the beginning of the conversation. I think it was Clara was saying that we have to take this to the lab now. What we got from here, keep in mind that this is an issue, keep thinking about it. That's the first step. Keep thinking about it. And then thank you all. Thank you for your presence, your discussions, your presentations, your questions. And we'll keep discussing about this.
Thank you for the initiative as well and the organizing.
We probably didn't solve the problem of cancer reproducibility today, but it's the beginning.
Close enough.
We're gonna move forward. Thank you all for, and we can keep discussing with coffee.